Okay, so we're back to molecular effects. This is just a drawing that shows the different types of things for macromolecules. The macromolecule we're mostly interested in is what? It starts with a D. DNA. That's what we really worry about. So you've got irradiation of DNA. You have what we call main link scission, which means just cutting, right? You have cross-linking, so these molecules form connections where they shouldn't, okay? And then point lesions, where you've got something that kind of interrupts the general appearance, the general construction of that part of it. Maybe it's been repaired, but it's like Legos that kind of look like they fit, but not quite, but maybe it's close enough. Okay, so those are the three types. Another view of molecular effects. So basically, DNA is transcribed. So you have messenger RNA, and then you transfer it to transfer RNA, which then translates it into a protein. And at any point in this is where you really don't want things to be broken apart. I would say you don't really want things broken here either, because transfer RNA is one piece that then combines with another one, right? So if you've got one that's good and one that's kind of like, when they try to combine again to form that DNA, it doesn't quite work. So S phase, DNA separates like a zipper. I love that analogy. You got two daughter DNA molecules are formed, each alike and each a replicate of the parent molecule. So that's the molecule we're involved with. So if DNA is the target molecule for radiation damage, it forms chromosomes, controls the cell, and the cells control human growth and development. Now, if anything interferes with this process, like radiation exposure or chemical, there's a lot of things that can interfere with this. You can have a lot of things. You got normal DNA. You have, this is what you'd see with what they call a terminal deletion. So it's been broken, and these are free pieces that just kind of float around. You've got dicentrics, so you've got pieces that combine. So you've got these two areas of connection. When this splits apart, it can do that. When this tries to split apart, it can't. And then you have rings. I've got some examples of those. So those are the different types of molecular effects. Back to radiolysis of water. Important. And I'm sure you'll be bored to death with this, but water gets irradiated, have ions, free radicals. In chemistry, the thing you don't want in a reaction are free radicals. They are violently aggressive. Okay. They are, I always consider them when I took organic chemistry, they're angry. They've had something stripped away from them and they become very reactive, and they will go out and find any atom, any molecule they can and grab. Okay. Very damaging. Now, what's interesting in radiation therapy, they want to create these things because these are what's going to go after the cancer cells and rip them apart. It's interesting, you know, a lot of people take things like vitamin C and things like that because they want to keep free radicals out of their body, right? When you have a patient undergoing radiation therapy, the first thing they tell you is, they ask, do you take vitamin C in any form. Well, yeah, I have orange juice, I take a vitamin C supplement. No. Vitamin C is not allowed during your course of radiation oncology because they want those ions floating around to grab onto those cancer cells and rip them apart. They don't want vitamin C or other things out there scavenging these things and taking them out of circulation. Target theory. Let me get through this and we can take a short bio break. There's a lot of different things that can happen. You can have a organic molecule can become reactive free. You can have, first of all, you can have hydrogen peroxide formed. By the way, that normally happens inside your cells. Part of the Krebs cycle for how energy is produced in your cells, one of the byproducts is hydrogen peroxide, as well as some other toxic things. Things that if you were to drink it out of the bottle, it would kill you, but your cells produce nasty byproducts like that all the time. So what you need to think about is your cells are constantly being bombarded, not just by radiation, but by their own systems of what they make. That was one of the things that led them to, repair. So if cells are repairing the things that they produce that are not good for them, maybe they repair radiation, and they do. So you can have organic free radicals formed in different ways. They're energetic, they have a unique structure, and the energy can be transferred to DNA and it results in bond breaks. In target theory, which is going to be next, cell death occurs only if the target molecule is inactivated. DNA is the target we talk about. It's in the cell nucleus. Okay. And that's when you talk about target theory. That's what we talk about. Let's take a 5-minute bio break, come back. We will finish the lecture, do a very brief in-class exercise, and then you can all go home. Oh, I brought stuff. Dun, dun, dun.
Nice.
If anybody's interested in food.
Thank you.
We have cupcakes.
Somewhere else, or?
Yeah, you can put them any place you want. Dun, dun, dun. Just something a little low-cal.
Read, that's yours.
Oh my gosh. Yes. Right back.
Yes, 2 of them are red velvet cupcakes, and then there's vanilla for those of you that don't like it.
I'm gonna be shy.
So we went to a dive bar nearby to watch.
That's what they're for. I can't take them home.